University de Utah, nos Estados Unidos. A palestra dele vai ser intitulada Color, Options and Diversity.
All right, bom dia. Thanks, that's all I know, right? So the rest is in English. But thank you for having me here today, and this has been really cool. I've enjoyed this so far. So I'm going to talk to you today about a project that I have going in my lab that is, it's not fully developed yet. You've all been presenting results that you have clear ideas, but you can kind of see a project that's kind of in the middle. And we're headed towards more of the ethology side of things, but right now we're trying to establish just some basic understanding of color evolution and visual system evolution so you can kind of appreciate where we are. This is work that I'm doing with my PhD student and my postdoc at BYU. So we all know how light interacts with the objects around us, and that light, when you get the full presence of light, it hits a surface and whatever is reflected is the color that you see. Pretty basic, right? And so this is how we get this whole array of colors where you have a full spectrum of light and then different lights reflected and different combinations of light leads to different color. And this is really interesting when you put this concept in view of biodiversity and what we see. So we know that color can lead to lots of different diversity. We see this in birds, we see this in fish, and we're starting to see it in insects more clearly, although we don't quite understand how it works there. So when you're looking at color, the next thing you want to look at is how those colors are received by the visual system. And when we look here, we know that humans, for example, us, we have 3 proteins in our eyes that allow us to see color, right? So we have 1 in the blue, and then we have 2 in the long wavelength, part of the visible light spectrum. Unless you're colorblind, then you only have 2. All right. But insects, the ancestral insect had 3. We'll talk more about that later. But we know other groups of organisms, like birds and a few others that we'll talk about, have more than just that basic 3, or some even less. But when insects see color, so we as humans, we see a butterfly like this. But insects probably see something more like this, where the UV pattern is stronger. And actually, their eyes don't see as clearly as ours. So an insect probably sees more like this compared to what we see, okay? So when we look at how color may have shaped diversity in our world, a group that I'm most familiar with, some research that I participated in were with heliconius butterflies, which you have here in Brazil, very beautiful. What we did is we went in and we measured the wing, the coloration in the wing. And what you see is that in heliconius butterflies there's a strong UV signal, and that the yellow forms a very distinct pattern that you don't see in the yellows of other insects here, right? And this yellow is part of a chemical called 3-hydroxykynurenine. And when you map the evolution of 3-hydroxykynurenine on the phylogeny, it's mapped here. And when you look at the visual system, the ability to see that color evolves right here, just in heliconius. So we think what we've discovered is the first example of a private channel of communication among insects. And it's pretty cool. It's the best example known among animals, but I'm biased too, right? It's my research. So, does color drive species and genetic diversity? These are the questions that we're trying to ask in my lab. And we use dragonflies and opsins. So opsins are the proteins in your eyes that allow you to see color. We'll come back to those in a little bit. So we have 2 projects that we're trying to tackle simultaneously. Color evolution in dragonflies and visual system evolution in dragonflies. So a little background on dragonflies. It sounds like you guys know dragonflies pretty well, right? From all the talks and from Heiner's work, Professor Heiner over here. It's good. But the order Odonata has 39 families and about 6,200 species. Okay, you have the Zygoptera, which are the damselflies, kind of skinny, wimpy, right? And then you have the dragonflies here, which are the more robust robust animals. What's really cool about dragonflies is they have really extensive color. Okay, the color's found across the whole group, and there's even the hypothesis that they may have been the first multicolored insect because of where they are in the phylogeny. They represent an ancient lineage, so that's kind of cool as well. So there's lots of great examples of color across odonates, but one of the more popular ones that's kind of gotten a lot of momentum in the research is this idea of sexual dimorphism. So you have a male generally just has one color, and then the female can have multiple colors. This is in a group, it's shown best in a group of the pond damselflies. There's more than 1,000 species. There's lots of coloration. You have female polychromatism, which is this. It's all throughout the group, but we don't know whether that's an ancestral state or whether it was evolved multiple times. And we have these questions of what's driving female polymorphism. There's different examples where it looks like it might be sexual conflict driving it. Other examples where it may be the ecology, an ability to deal with the sun's rays, or maybe even a combination of both. So using Odonata, it's this really great opportunity to just ask a very simple question. Is color a driver of species diversity? So in order to do this, we went out and we had to measure color. And measuring color in odonates is really hard, okay? Because they're small, right? The animals themselves are small, but the areas of color can also be very small, which is difficult to measure. I see some of you guys nodding your heads, trying to do it, right? The color fades and it varies, so after death, even if you preserve them well, the color can fade and change. Temperature has a fluctuation in color, right? Even when you get mad at them, the color fades, right? They're like little children, you know. And then we have two methods that we've used to measure color that kind of help us with this difficulty. So we have what we call the clean method here, where we use reflectance spectra with a probe and we can measure the actual reflectance of light on a live animal or a newly captured animal. We use these different tools to measure it. We measured 4 body regions for about 232 species, mostly from Africa, right? And that it represented about 1/3 of the families. The other method we call the dirty method, right? And this is the method where we just measure from images, and we've measured 8 body regions for about 850 species and all the families. And I'll show you some results here. So this is the clean method. This is the better method to measure by. And what we notice is there's 2 major peaks. You have one here in the green, one in the long wavelength, and very little in the UV, which is less than expected. We thought UV would be a very important signal for odonates, but we're not seeing that in the measurements we've done so far. But we've only measured 232 species, right? You know, there's almost 6,000 more to measure. So, and then as you look down here, you have this minor peak in the blue, right? And so I'll show you the dirty data now, right? So this is the dirty data. It's mapped in a circle because that's how you map hue values. And you can see there's lots of similarity between the two, maybe just a little bit of a shift. But this allows us to go in with the dirty data and we can look specifically at the colors in every family. So this is the family Eschnidae. There's about 500 species. You can see the color distribution in here. And this is Petulurti with only 13 species and the color distribution here. And since we've gone in and we've measured color across all these families, we can now ask the question, is there evidence that color might be driving species diversity? And I'll show you our first evidence. So if you look here, you have species diversity on this axis and what we call a Hellinger distance on this axis. A Hellinger distance is just the difference between 2 distributions. Okay, so we have the distribution of all odonate color and the distribution of each family's color. And when you compare those, you get a distance. As you can see, the most diverse are the most colorful, all the way down to the least diverse, or least colorful. So we have this first compelling evidence, right? It's not anything set in stone. We call it smoke. It's smoke instead of fire, right? But we can see that there's some evidence that diversity is linked with coloration. And when we've had Diogo's talk here and others, it starts to make sense, right? The odonates are using color as an interspecific and intraspecific signal to see each other, and it may have actually driven diversity in the group. Pretty cool. So A little bit of breather just to review. So we have two major peaks in the color of odonates. There appears to be highly significant correlation between diversity and coloration. And then this allows us to ask these other questions, right? So we'll now that we've kind of established that, we can move on. Say, can color also contribute to genetic diversity? So we think it might be driving species diversity, but what about genetics? Okay, if it is, what about the genetic diversity of the visual systems? You would expect that the genetic diversity in the visual system would be diverse as well, right? If they have to detect that color, you'd expect diversity there. And then that's kind of what we're talking about here, but specifically we're going to look at these genes called opsins. So real quick, I'm just going to review genome. I think you all know what a genome is, no problem there, right? So genome, you got the full, if this blue circle represents a genome, the full circle is all genomic data, right? All DNA material, okay? And then you go down to the exome. The exome is just all the exons in the gene. This is in the genome. This is all the areas of the genome that could potentially be translated, right, transcribed into a protein. And then you have the transcriptome. And the transcriptome is what we'll talk about today. And it's a little bit more nuanced, or a little bit more simple, right? This is all RNA transcript in a given tissue at a given time. And it's a little bit more complex just because there's variation due to development. So whether an odonate is a naiad or an adult, the environment can play a factor, time of day, tissue type, right? You're going to get very different transcriptome if you pull from the muscle or if you pull from the eye. Okay, so we're messing with dragonfly transcriptomes of the eye. All right, and so if you look here, what we think we have here in odonates is like this pure system to look at evolution. There's not much evidence. There's a little bit of evidence that they use their antennae as adults, but very little. It looks like the major sense that they're using is their eyes. So we have this system that we can go in and look at the evolution of visual systems that isn't being compromised by other senses like touch and pheromone, etc. All right, so phototransduction. That's vision. That's a big fancy scientific word for vision, right? Okay, and this is where you have the opsin gene, which is shown here in these pillars, how they interact with the chromophore, which is this internal structure here represented by the R, and together those are called a photopigment. Okay? So what's really cool about this group and studying opsins and photopigments is that we have a pretty good understanding of how opsins and photopigments work together. We know the structure, we know the function, spectral tuning sites, we know specific amino acids that will shift the vision one way or another in sensitivity. And we know a good amount about the general evolution of the group, so it's a good gene to work with. So back to this, right? So just a quick review. We know that we have the different phototransduction pathways here, and that insects can see in the UV where we cannot, which is unfortunate. Wouldn't it be cool if we could? Make things a lot easier, right? And then, so the questions we are asking specifically is, what are the diversity of opsins of dragonfly visual systems? So how diverse are their opsins? What are the patterns of selection surrounding their opsin evolution? Now remember, the ancestral odonate had the ancestral insect had 3 opsin copies. And we started to look around in the literature at this. And so we took transcriptomes from the eyes. So we had 2 outgroups and 16 dragonflies that we looked at. And this is a lot of methods that you don't need to worry about, right? But it's just kind of like how we went through and pulled the opsins out of the transcriptomes and were able to look at them. If you have any questions about that, we can talk about it later. But as you get into the sensory systems, if you look in the literature, dragonflies, if you look at physiological data, they estimate that they have 3 to 5 photopigments in the eye. That's what they're guessing. But in damselflies, only 3 photopigments are estimated. There's no other physiological data that would show more than that. But when we looked at the transcriptomes, when we sequenced what was found in the tissues, we found 10 to 30 opsins in dragonflies. And we found 9 to 22 in damselflies. And if you look, we only found 1 ultraviolet, 3 to 8 blue sensitive opsins, and 9 to 21 long wavelength, and a very similar pattern in damselflies. So pretty cool. This is the most diverse insect that we know of so far in terms of visual system. One of the most diverse animals in the world in terms of color visual system. And so we started to look around at other groups, and we looked at stomatopods, right? So stomatopods, they have a lot of opsins as well, but the physiological data aligns very well with that, right? So if you look at the physiological data, people estimate they have anywhere from 15 to 20. When you do the sequencing, you find 15 to 20. But you don't find that in dragonflies at all. So why is that? It seems really strange, right? So what's going on in Odonata with their visual systems? We don't quite know yet. We're still digging. But let me give you a view. So if you look at opsins in a phylogenetic context, you can see that these are the photopigments here. You can see very little, only one UV, lots of gains and losses, right? And if you look at a larger one, this is ultraviolet blue, and then this massive expansion in long wavelength. And this is another view of it on a phylogeny. You can see this is the ancestral state of 1 ultraviolet sensitive, 2 blue, and 10 long wavelength. And then you can see the gains and losses as you go throughout the phylogeny, across the tree. Pretty interesting. But one of the things that we've kind of looked at with this is this idea that if you remember back to the color data that I showed you, ultraviolet wasn't providing much of a signal. Blue gave us a minor peak, and the long wavelength is where most of the color was. And it seems to align really well with what we're finding in the visual system. So again, some smoke. Not a lot of fire yet, but some smoke. So when we went in and we looked at the evolution of if we looked at selection on these genes, how much they're actually evolving or pressures that are pushing them to evolve. One of the things you can see is if you compare odonate opsins to all other insect opsins, there's more relaxation of evolution on these opsins than in other insects. And then you have the ultraviolet, the blue, and the long wavelength out here. So what we're actually showing in this graph is that the long wavelength is allowed to evolve much more than the other genes in the eye. And this is another view. This one's a little bit complex. I'll try and explain it. But essentially what you have here, this is blue sensitive opsin and long wavelength sensitive opsin. What you have is, if you look at the phylogeny here, you have a duplication. So you have a duplication here, and a duplication here, and a duplication here. This is before the duplication, and then this is after the duplication, and then this is when the duplication is set. And what you see is the amount of selection decreases over time. All right, so I'll kind of wrap this up. I would argue that odonates are a model system for opsin evolution, and if you guys have any interest in this, you should get into it. There's plenty of room. Everyone can play here, right? And this work would not be accessible without genomic tools like transcriptomics, because they have so many copies, it's impossible to do enough PCRs to find what you're looking for. But beyond that, I think opsins are this really cool playground for evolutionary ecology and genomics and behavior even. I think they can help explain a lot of things. So in future directions in our lab, we're looking at genomes to look at how the opsins are distributed in the genome, how they're duplicated, how they're evolved, how they're selected for. And then we're exploring evolution of opsins within Eschnidae. So this is Eschnids here. These organisms have the largest compound eyes of any arthropod. They have extreme opsin diversity, and they're highly colorful. And then some are not. Some are just pure brown, and some are extremely colorful. So it sets up a nice comparison for us. And then we're looking deeper into the relationships of color and color vision. Like I said, it's really smoky. We don't have a lot of fire yet, but we're working on it. So these are the funding sources who provided. This is Anton, my PhD student, Millie, my postdoc. She likes putting stuff on her head. And then I'll leave you with this picture. So this is a pseudostigmatine from Central America. There's a spider hiding up in here. It's about to eat lunch. I think we are too at some point, right? So with that, I'll leave you with any questions.